Okay, so we're now going to look at the light independent reaction, which is sometimes referred to as the Calvin cycle. We've got the ATP and the reduced NADP or NADPH from the light dependent reaction, and that goes to provide reducing power and energy to convert or reduce CO2 into glucose ultimately. And that occurs in the Calvin cycle. Light at the top there is trapped in the light dependent reaction and ATP is produced. Also, the hydrogen from the splitting of water at the bottom is loosely bound to the NADP to become an NADPH. Both of those go to be spent, for want of a better term, in the Calvin cycle, and ultimately CO2 will become glucose. Not straight away, it becomes GP first, but ultimately it'll become glucose. So it's named after Melvin Calvin, who was an American scientist who actually discovered the cycle. He went and did a very famous experiment, the lollipop experiment. It all takes place in the stroma, and what happens is that ATP and reduced NADP is used to reduce carbon dioxide into sugar or glucose. It does not require light, so that's why it's the light independent reaction. And as I said, it was the American that discovered it, and he put his name to the cycle. The cycle itself is very complex. We start with RUBP, a 5 carbon molecule, which CO2 is added to, and it becomes two 3 carbon molecules. So we have 5 carbon plus 1 carbon equals 6, but it gets split in half into two 3 carbon molecules. The ATP and the reduced NADP from the light dependent reaction then not only converts that, but also reduce that into TP, triose phosphate. And because there was 3 carbons within the two molecules there, there's 3 carbons within the two molecules there. Using a further bit of ATP, most of that gets regenerated back to the ribulose bisphosphate or RUBP. But for every 3 turns of the cycle, a glucose molecule can come off. This is actually really complex, so hopefully I'll be able to explain this through a diagram later on. But just in principle, remember that it's RUBP gets converted to 2 lots of GP, which gets converted to 2 lots of TP, which then can be regenerated back to RUBP, and also some useful substances such as glucose. It's a little bit more complex than just it's shown there. Here we've got 3 complete turns of the cycle. We've got in white there 3 lots of RUBP. That joins with 3 lots of carbon using the enzyme Rubisco, which is a vital enzyme that powers this reaction. It allows the conversion of the RUBP and the CO2 into GP, which is in green at the top there. That GP is then further converted using ATP and the reduced NADP into TP. Most of this, in fact, all of these bar one, will be regenerated back into the RUBP there, ribulose bisphosphate. So if we count them up, there's 3 times 5, so that's 15. And there's 5 times 3, which is 15 carbons. So most of the carbons go there, but for every 3 turns of the cycle, 1 is left over. Hopefully this will become more obvious in a minute when I go to my picture, but just accept it for now, it's 3 turns of the cycle. So what we've got here is my simplified diagram of RUBP. You can see it's 5 carbons long, and it's a holding molecule. What's going to happen is we're going to add to this as we go along. The first thing we're going to add is CO2. So we've got to take the CO2 and add that to this. CO2 has 1 carbon in it. So what happens is the RUBP, using the enzyme Rubisco, really important enzyme, catalyzes the whole reaction, will take the carbon in CO2 and add it to the RUBP. And ultimately, we will end up with 2 lots of GP. So you can see there's 6 carbons there, 2 lots of 3. This then gets converted using the ATP from the light dependent reaction and also the reduced NADP from the light dependent reaction. And both of those use their abilities, energy and reducing power, to convert the molecule into 2 lots of TP. Here, most of those gets regenerated back to RUBP. But for every 3 turns of the cycle, we end up with 1 spare molecule being left over. I'm going to draw all this out in a second, so hopefully it'll make a bit more sense when we see all 3 together. So I've neatened it up a bit here, and what we're going to do is we're going to add some more molecules to this. So that's 1 complete turn of the cycle there, which is not what happens. We need to add 2 more turns. So I'm going to do it in another colour. So what we do is we're going to add the blue RUBP here. That's 5 carbons. And we're going to add another CO2, and that's going to make another 3 GP here. So 3 carbon GP, so 2 lots of that. That is going to be converted by the ATP and the NADP, which I've removed just for sort of ease of access. And it's going to be converted into the TP here. We're still not quite finished yet. We've only done 2 turns of the cycle. So we need to now include another turn, which we're going to do in green. So again, just bear with me. We've got 5 carbon RUBP plus 3 carbons now from the CO2 equals 18 carbons total up here. Another 3 added to that, so that's 18 total. I'll write the carbons on in a second. They get converted with the ATP and the reduced NADP into another lot of TP. And now we've got 3 turns of the cycle going on. So it's 3 turns. So we had 15 carbons plus 3 carbons equals 18. Those 18 get converted into 18 here. And you can see 15 of them get regenerated. So what we can do is we'll take 15 of these. So I'll just highlight those in red. There you go. So all those get regenerated back round. So all those are just sort of going round in a circle. But this spare one at the end here in green is going to be left over. That can leave the cycle. So I'm going to convert it back to green here so you can see it. There you go. Now, what you've got to imagine is there are millions of cycles going on in the stroma all at once. So it's quite logical to say nearby another cycle is going on. So we've got another cycle going on down here in this region, 3 of them, which has made another TP, which is spare to the system. Now, this is where the real magic happens. If we bring these together, hopefully you can see we've just made glucose. And that is the stable molecule that can leave. TP is relatively stable. So 3 turns of the cycle will make TP, stable molecule that can leave. 6 turns of the cycle can make glucose. But we go, well, TP is stable, so therefore that's the one we're going to focus on. So a couple of ways of imagining this. One way is imagine someone standing there with their arm out and turning their arm in the air 3 times. So it's one person doing this with their arms 3 times. So I'll just draw the person on, there you go. So they're sticking their arm out and they're drawing it in the air 3 times. That's not what happens. You've got to imagine 3 people standing side by side, and each one is waving their arm in the air once or circling their own arm once. So this one does it once, this one does it once, this one does it once. Together they've each done 1, but between them they've done 3 turns of the cycle. Or another way of imagining it is a bit like 3 London eyes side by side. You've got one in the middle, and then either side of them have got 2 others. They're all going round simultaneously. And what happens is a person steps up to the first one, the second one, and the third one simultaneously. They represent the carbons in CO2, and they step onto the gondola of the London Eye and start to head round in that sort of cycle, because they're always moving very slowly, so they're going round. And they'd be termed in Facebook and other social media as being in a complicated relationship. So they kind of, the one in the middle looks left and right and reaches across the void of space, and they take hold of each other's hands. So they then sort of reach across the void, hold each other's hands, and then step off the other side of this little love triangle, which is virtually identical to how the TP is produced in the Calvin cycle. This little triangle of 3 carbon atoms, they started as individuals, they went round the cycle and came out the other side as this little triangle TP.